for a moment here. And I think we're all back. Thank you
Yes.
all.
Thank you, Philip. This is fantastic. Really, this is the brave new world of Huxley's brave new world of adipocytes. It's fascinating, the discoveries that you have made and the fantastic translational impact that such discoveries have had and will continue to have. With that, I'd like to acknowledge our panelists. I have Professor Dr. Tagmeyer joined us, Dr. Laurie Kirsch. Oh, Dr. Tagmeyer, here's his video. And Laurie Kirschenbaum is as well had joined, and Yibing Wang, and we have also I see Priyantash Gurha. He should be on the panel as well somewhere. He is. All right. With that, the podium now, this presentation open to question. I would like to ask one question about endotrophin. I wonder whether it targets fibroblasts, does it transdifferentiate them to adipocytes? Is a transcriptional switch between adipocytes and fibroblasts, or it just simply shuts down profibrotic genes and overturn the collagen turnover, and then fibrosis goes away?
So we still don't fully understand the signaling behind endotrophin. We are still looking for a receptor that we have not yet identified. Downstream signaling seems to involve a lot of the classical TGF-beta SMAD pathway. But what happens basically is that when you neutralize the endotrophin, much of that TGF-beta-like signaling disappears, ceases, and as a result, you kind of wait for the turnover of these collagens in the ACM, and at least a partial resolution of that occurs.
The reason I asked that question, we have a heart condition called arrhythmogenic cardiomyopathy. In a subset of them is one of the very unique phenotype is enormous infiltration of adipocytes into particularly the right side of the heart, not exclusively. So I was wondering and I think endotrophin might be a very good, is a fibroadipocytic infiltrate. I was wondering potentially could be a good therapeutic answer to this condition. So I was worried whether if you target adipocyte, you will have fibroblasts, you will have more adipocytes. If both of them go away, of course, that will be fantastic. Other questions? I see Laurie's hand is up. Laurie.
I'll jump in. Dr. Scherer, that was a brilliant talk. Very, very elegant and so nice and detailed, yet so simple. What I wanted to ask you was very much following up what AJ asked about the endotrophin. You were mentioning that it's a cleavage product of collagen 6. So is there a specific protease? Has that been identified, or and is that activated under more stress conditions that would generate more endotrophin?
Yes, so that's an excellent question. Work by Kai Sun actually in Houston has revealed that MMP14 seems to be one of the proteases that's particularly prone to cleave endotrophin off. It is very efficient in terms of cleavage. People frequently ask, would that be a possible site of intervention when we prevent MMP14 activity? We've never really seen endotrophin being part of the parent molecule. It seems to be very efficiently cleaved off and has a life of its own in this particular context. MMP14 does go up in the obese state. Overexpression of MMP14 in fat cells leads to higher levels of endotrophin. So it is an integral part of the biogenesis of endotrophin, which can be produced in the mature fat cell, but it can also be produced, of course, in myofibroblasts, macrophages, whatever eventually infiltrates these profibrotic lesions that you see.
It reminds me very much of endoglin. And when you talk about the TGF-beta signaling pathway, that a lot of these products are formed because of a large parent that then ultimately becomes clipped, and then the children, if you will, the offspring, have their own biological properties, which is really quite interesting. Thank you very much for a great talk.
Appreciate it. Thank you, Laurie.
Excellent. Yibin has a question posted online, and Dr. Tagmeyer is waving his hand. I guess we got to give the priority to Dr. Tagmeyer, Yibin.
Thank you very much, Phil. That was phenomenal, and always, you're opening a new world and opening new perspectives, and everything is new and yet fits together. One of the more straightforward and simple aspects that I may have missed is the effect of diet, and especially de novo fatty acid synthesis from carbohydrates by the liver. In the setting of obesity, and of course, then in context with the GLP-1 receptor story and the endotrophin story. Any comments? Because that seems to be, at least for our patients, seems to be a very big issue.
Yeah, the association, of course, with fatty liver disease and whatever ensues with the steatosis, with the enhanced de novo lipogenesis occurring in the liver, with the enhanced flux of free fatty acids from fat tissue to the liver, all leading basically to more steatosis, more fibrosis, is, of course, a major point. The effects of the dual agonists on energy expenditure, but more importantly on food intake, are obviously very relevant. So what you see in these patients is basically a caloric deficit for the most part. And with that, your fatty liver and the fibrosis sort of melts away. You have enhanced insulin sensitivity as a result in the liver, and as such, reduced levels of de novo lipogenesis as well. So I think much like statins take care of the hypercholesterolemia, but there is a residual risk even post-statin. I like to refer to the post-dual agonist world that will have residual risk or residual phenotypes in the form of leftover fibrosis, even after the steatosis resolves. That fibrosis can disappear in some patients, but it will not disappear in others, and hence there is still going to be a demand to effectively intervene at the fibrosis stage as well.
So, Phil, how do the dual agonists compare to non-pharmacological interventions like bariatric surgery?
There is a lot of parallels actually between bariatric surgery and the dual agonists. In fact, the excitement about the weight loss of that magnitude, 20%, 25%, whatever it is actually mostly based on results from bariatric surgery studies, of course. We know some long-lasting studies in the context of bariatric surgery. The Swedish SOS study really revealed what that amount of weight loss can actually do. And it really stretches across the board from diabetes, cardiovascular disease, and cancer. Cancer risk is reduced post-bariatric surgery. Now, the advantage that...
And heart failure. And heart failure.
And heart failure, absolutely. The advantage, of course, that the dual agonists offer is that it is less of an intervention. I think it's fair to say that while there are certainly side effects of these drugs, they are well within what people consider tolerable. There's mostly a little bit of nausea initially, but I think the less the nature of the intervention with the pharmacology versus the surgery makes the pharmacology seem more attractive. And as such, the future of bariatric surgery, the bariatric surgeons don't like to hear that, but I think there will be fewer bariatric surgeries performed as we progress towards a more widespread use of these dual and triagonists eventually.
Less painful for sure.
Yep.
Wonderful. So we have Yibin, you get the last say.
Thank you so much. I really appreciate the opportunity, and it's amazing lecture. I learned so much. And actually, you already addressed my first question, is how this endotrophin actually being produced independent actually from the collagen 6-3 alpha, sort of the maternal gene expression, as you indicated. I just wonder, because based on the expression profile from our own study, it's also highly enriched in the fibroblast. And I wonder whether this molecule also play a very important role in the wound healing, the normal wound healing process. As a result, you would think about how to utilize the neutralizing antibodies for post-MI or versus the sort of the long-term remodeling aspects.
Well, very important point, Yinbin. That is was actually one of our concerns for the neutralization. So what is the actual role of endotrophin? Why do we have endotrophin? What does it do? It's got to be there for a reason. And so far, we have not yet found a functional role for endotrophin under any pathological conditions. Endotrophin knockout mice, collagen 6 knockout mice seem to be relatively normal. They don't have any major phenotypes. There's something called a Bethel myopathy, which is a very low level myopathy, but that's because they're missing the full fibrillar collagen 6, not endotrophin. So right now, endotrophin falls into the same basket as PCSK9. We don't know why it's there. All we want is neutralize it, basically. We have no evidence at this point, and others have studied this a little bit more extensively. There is no evidence for an involvement in wound healing. So that was our first go-to thing as well. Would a neutralization of endotrophin potentially impair wound healing? And that does not seem to be the case so far. So we're still looking for a normal physiological role of this molecule.
That's amazing. And as a follow-up, it's a question about the the GIP receptor transgenic mice. Considering such a high level of basal energy expenditure, just from a philosophical point of view, what is the impact on their lifespan? This is actually a journal for cardiovascular aging, and we always feel there's a lower energy expenditure is preferable or beneficial for lifespan or health span.
Yeah, good, excellent point. Is that actually a bad thing if we ramp up the energy expenditure that much? What's actually remarkable with these mice is that they will completely normalize their food intake, but they're much skinnier. So by our definition, depending on how you normalize, they either have normal food intake or they're actually hyperphagic. And they expend that energy. And I can't answer the question for longevity yet. I mean, we let these age now and see what happens, whether that has an impact. But there is a very significant increase in energy expenditure. Absolutely, you're right. And I don't know whether we should be concerned about that or not, but whether or not that is associated with an enhanced, aging process remains to be seen.
Yeah. Fantastic, beautiful, enlightening day. Enjoyed it. Thank you so much, Dr. Scherer. This was really a tremendous day for us. Wonderful educational opportunity, and grateful to you for your fantastic presentation and marvelous scientific discoveries. Have a beautiful day, everyone. Thank you.
Thank you
All. Appreciate it.
Thank you.
Thank you.
Bye-bye.
Bye.